What equipment is that?
Recording. Special recording equipment for focus groups. I'm like, okay.
He just don't do that. So
Oh, this, okay. I'll write that down. Yeah. Yeah, it's kind of a complaint from a lot of people I've heard. For me, it's just about as convenient. Yeah, you could drive it. Maybe the 8 hours probably. But it's a hour. Yeah.
I was told next blogger.
Turn these around so people can know what this is.
Florida's always like Phoenix is big. It's usually not terrible. Yeah. Well, Southwest and US is my spot.
I want to make sure people know what's here.
Actually,
Yeah, yours straight up.
my sister's right here.
Tips are small. It must be extra long.
Birth starts now.
no, that's
Well, Barbara said, you set up your team?
I can't even get one.
I knew that. I knew that was going to happen.
Yeah.
Whatever you want.
They got us kind of framed in here.
Whatever you do, let's all right.
How's it going?
She's fine.
He's good.
Keeping busy.
I don't know if it's a good thing or a bad thing that he's going to
Guys,
He
like
asked me to say hello to everybody.
the other way.
Yeah.
Yeah. I think we have 11. The goal was to try to get 15 people. I think we were 11. Maybe. I guess the advantage
They get vegan, so
is extra.
yeah.
And the merchants doing it for years.
Were you married?
So she would do this states. But it should be, I think they made it more interesting. So we actually go through the
Yes, there was some odd things I saw. Yes, a little bit of oddness in the way that they do things, but
like an hour and a half lecture.
Yeah, no, which is right. So there's right up in the next so we had about how many years? 65.
How many years? Way less people started.
Driving around
The patients and letting us
You
do
got bad cough.
electrophysiology.
going to see triage.
Jeez.
So it'll be that they're
Gift of
That's
the
the
Yeah.
just the rest
Air trip,
of
yeah.
Oh, jeez, yes.
It was,
You do much medicine anymore?
I was interested in bioparticle function.
Mine addiction.
It's so true. Never say anything near a microphone that's so unfair.
Okay, good morning. We're going to get underway. If I can get your attention, I'd like to get the program underway. The medical panel is here. These are the members of our medical advisory board. They're here to discuss the research that they're involved in or other research that they may be doing in the future. And this is a unique opportunity to hear from many of the top experts in the myositis field. Some of you may know our medical advisory board is an unpaid group of volunteer physicians who devote their time and energy to not just helping you, but helping TMA. They review our research proposals that we receive. They recommend to our board of directors what proposals are worth funding, and then our board of directors makes those decisions. They also go out to medical schools, talk about myositis. They try educating young residents and interns and getting others interested in this field. As you know, rare diseases are not at the top of the list when students are going to medical school. If they're lucky to hear the word myositis, that's probably the exception rather than the rule. But we're doing a pretty good job of getting to the medical schools now and trying to get myositis on the radar. And these are the folks who are doing it. They do a lot of other things to support the organization. So I'm going to turn the microphone over in a minute and let Dr. Wortman, who's on our board of directors, moderate this panel. But I did want you to join me in giving a round of applause to our medical advisory board. Thank you. So a couple very brief announcements. The silent auction, if you haven't looked, you ought to. It's to the right of the registration table outside the doors. The auction's going to close at 12:45. There's actually about a dozen different items out there. Hopefully, if you're interested in bidding, you may want to check because there's been a number of bids this morning. So if you are fortunate to win, you'll find out at lunchtime at 12:45. That's when the sheets will be picked up. I mentioned the dinner this evening in the Capri Ballroom, 6:30 to 8:30. It's one of these dinners where there are food stations. You can come in anytime during those 2 hours, sample foods from 4 of the restaurants in the hotel, and really, again, it's a beautiful place, great opportunity to have a nice meal and spend time with your colleagues and share information about the disease. So tickets are $45. They're still on sale. Go to the registration desk. We're really not wanting to sell tickets at the door. So if you're intending to go, make sure to buy your tickets probably before about 4 o'clock this afternoon. We're going to have to give a meal count to the catering staff at that time. The vendors, again, if you haven't had a chance to stop by, please let them know we appreciate their support. The last item I want to announce is I mentioned about a focus group for dermatomyositis and polymyositis patients. We have about 13 people who've signed up for that. They're still looking for more, but they are going forward with those 13. Dr. Lisa Christopher Stein and Helen Alexanderson. Helen's over here. They're the two who are going to be working with the focus group. Those who've signed up, the 13, when this session finishes, if you go outside to the registration desk, they will meet you there and take you to where the focus group is taking place. If there's other DMPM patients that want to participate, as long as you're not a patient of Dr. Stein, you're eligible to be part of the focus group. So with that, I'm going to ask Dr. Wortman to take over here. I think, just so you know, we're going to try letting all the members of the panel have an opportunity to speak before you take questions. There will be a chance for you to ask your questions, but you're going to have to hold those until everyone has spoken. So, Bob?
Thank you. I'm Bob Wortman. I'm a member. You hear me now? Yeah. Okay. I'm Bob Wortman, and my pleasure to moderate this panel discussion today. I'm a former member of the Medical Advisory Board, and as I'm a rheumatologist and a professor emeritus from the Geisel School of Medicine at Dartmouth. But after serving on the Medical Advisory Board for several years, my term ran out, and I was able to join the board of directors to continue to work with this organization. So that's about me, and now we're going to hear from the rest of the Medical Advisory Board. We'll start at my left with Alan, and they'll just move from left to right till everybody's introduced themselves and told you a little bit about themselves, and then we'll open it up to questions.
Good morning. My name is Alan Pestronk, and I'm a neurologist at Washington University in St. Louis. My research is based on the idea that some of the ways we think about immune and inflammatory myopathies are in need of a little bit of renovation, like an older house. Dermatomyositis, for instance, is probably now 4 different diseases. So if the doctor tells you have dermatomyositis, maybe you should ask which kind. Polymyositis is a disease that some people say hardly exists. Half people with inclusion body myositis don't have inclusion bodies, so what disease do they actually have? And so what we're trying to do at Washington University is build a different kind of foundation on our house, where we're looking at muscle biopsies. I'm a pathologist, and trying to find ways of looking at muscle biopsies that more accurately define which diseases people have, what things they're predisposed to have happen to them in addition to their weakness, and hopefully to predict which kinds of treatments are most likely to be useful for them. And so that's the major focus of our efforts. We're also interested in understanding what the particular proteins or other things in muscle that the immune reaction is directed against, so we can figure out how these what actually causes these diseases. Thanks.
Good morning. My name is Christina Charles. I'm at UCLA Medical Center, and I'm an academic rheumatologist. The way I got interested in myositis is somewhat of a sad story, and I'll briefly touch on that. It was years ago when I met 2 women who were referred from the lung transplant unit who had end-stage pulmonary disease from the dermatomyositis, and they really hadn't been treated aggressively, hadn't actually been diagnosed for years. And so at that time, no one was studying myositis at UCLA, which is a big referral center, and I decided that's what I needed to do. I now have several very happy, successful stories to tell about patients with myositis whose lung disease has not progressed, who have done very well. And that's why I do what I do. In terms of, and we also, I should say, now have a fairly large center that's growing. We have referrals from outside California. And out of all 20 or more rheumatologists, most of them will call me if they have a case of myositis. And I think we're doing a lot of good things in the clinical setting. In research, we are building our cohort of myositis patients. I've had a special interest in the function of lipoproteins, in particular high-density lipoprotein, which is also known as HDL, the so-called good cholesterol. And this lipoprotein is important in protecting blood vessels. And certainly we've looked at this as a risk factor for heart disease in other patients, and now in patients with myositis. And we're finding some significant abnormalities. That may be important for heart disease, but it may also be important as a different mechanism for targeting blood vessels in muscle. And so that's the research we're pursuing at present at UCLA.
All right, good morning. I'm Andy Mammon. I'm currently at the National Institutes of Health, but I got interested in myositis as a neuromuscular fellow at Hopkins. And I think like you, I met a couple myositis patients and realized there was a great unmet need, and really no one at Hopkins, in neurology at least at the time, was working on myositis and specializing in myositis. So I decided to go that direction. And soon enough, I realized that there was another person at Hopkins who was really interested in myositis, who was a rheumatologist who's here on the panel, Lisa Christopher Stein. And so the two of us decided to do kind of a novel thing, which was to start a myositis center that included both neurologists, rheumatologists, and pulmonologists, kind of all sitting in the same room discussing the same patients. And we have collected quite a cohort of myositis patients and have together been involved in a lot of research. In part, we've been interested in trying to understand how certain environmental factors might trigger autoimmune disease, and have found that in some patients, a minority of myositis patients, statins appear to have triggered the disease. And we've been really interested in understanding the genetics and environmental triggers for myositis. In the last few months, I've moved to the National Institutes of Health, and that's where my lab is now. And the lab is focused on understanding, like Alan, I think, trying to define the different types of dermatomyositis and polymyositis, recognizing that there are probably multiple different diseases. We come at it from a little bit of a different angle, looking at the autoantibodies, and are interested in whether those autoantibodies identify unique populations. And in the laboratory, I'm also interested in understanding basic mechanisms of muscle regeneration that might be applicable to not just patients with myositis, but patients with all different forms of muscle disease. Thank you.
So good morning. My name is Helena Alexandersson, and I'm a physical therapist and a PhD from the Karolinska University Hospital in Stockholm, Sweden. I got interested in myositis in the middle of the 90s when I met my first patients, and when I started to look for recommendations on exercise and physical therapy, in the textbooks it said, well, you should stay away from active exercise and maybe do range of motion. And at that time I got to know Professor Ingrid Lundberg, who is a rheumatologist at Karolinska, and she had said to her patients, don't exercise and rest. And she had a couple of patients who did not follow her recommendations and did in fact do active exercise and improved markedly. So we started to think, well, maybe you can exercise if you have myositis. So we set up a couple of studies to explore, is it safe to exercise? And we got encouraged by our results that it doesn't seem to increase your inflammation or disease activity. It's rather the opposite, that it could be an anti-inflammatory treatment. So my research today focuses on to understand more about the mechanism of exercise effects in myositis. And we also, with the qualitative studies, are trying to understand more about the symptoms that you, as myositis, living with myositis experience, and also to develop patient-reported outcomes that are important for you to assess. Thank you.
Good morning. My name is Roop Tandon, and I'm a neuromuscular neurologist at the University of Vermont, College of Medicine. I became interested in neuromuscular disease when I did a fellowship with a gentleman by the name of Dr. Walter Bradley. And Dr. Walter Bradley is now retired, but he at that time was doing research in muscle disease, including in inflammatory muscle disease, and had written a very extensive review on patients with inflammatory muscle disease with a gentleman called Dr. DeVere. So DeVere and Bradley had written an important review. I run a fairly large MDA and neuromuscular clinic where patients with all sorts of muscle disease and nerve disease are seen, but also particularly patients with inflammatory neuromuscular disease. I'm involved in the training of residents and fellows, and we've trained many fellows who have gone into neuromuscular disease, who are doing research in that area. In terms of my interest in the research arena, I've been involved in drug development in neuromuscular disease for the last 20 plus years or so, and continue to do so. The other area of research is in examining some of the nutritional aspects of neuromuscular disease, some of the physical activity levels in patients with neuromuscular disease, and trying to quantify those activity levels in patients objectively, and also looking at energy utilization in people with neuromuscular disease using what's called mass spectrometry or stable isotope techniques, which are very sensitive techniques to look at energy conservation or energy utilization in people with neuromuscular disease. I hope to be involved, maybe use some of the tools that I have in patients with inflammatory muscle disease, including some of the nutritional aspects of the phenomena that happen in people with inflammatory muscle disease. Thanks.
Good morning. My name is Zarif Shaheng. I'm a neurologist, neuromuscular disease specialist, and the director of the Clinical and Experimental Neuromuscular Laboratories at Nationwide Children's Hospital Research Institute at the Ohio State University. My laboratory has been instrumental in analyzing the tissue in the gene therapy clinical trials and other clinical trials in the neuromuscular disorders to assess the treatment effects. I'd like to take this opportunity and tell you about give you an update on the follistatin gene therapy in Becker muscular dystrophy and inclusion by the myositis. Some of you may know that this study was supported by the Myositis Association. We're very pleased to announce that we have completed the Becker muscular dystrophy part of the study, and it is a positive study. This is actually the first study that we have shown functional improvement, and two out of actually four patients out of 6 showed improvements on 6 minutes walking test. 2 of the patients, this improvement was over 100 meter. The other 2 was a little bit more modest in 57 and 28 meters. The part of the study related to IBM was delayed a little bit because FDA asked us to generate some safety data, so we only injected one leg, and this was a low dose. But we did actually complete it. 3 IBM patients injected the high dose in both legs, and all of them did improve. One over 149 meters, the other 2, 62 and 23 meters, and we will go ahead and finish this study by enrolling 3 more IBM patients. So I thought I would share this information with you. Thank you.
Thank you. Good morning. My name is Chet Otis. I'm from the University of Pittsburgh. I'm a professor of medicine, and I also speak for Dr. Rohit Agarwal, who is on this board, but his wife is having a baby soon, so he'll be here next year. Together we direct the Myositis Center at Pitt. So I've kind of taken the lead here from Drs. Pestronk and Mammon. Dr. Pestronk being the neuromuscular pathologist, and I'd like to pick up on Andy's point, then we've been interested in autoantibodies as biomarkers of different subsets of myositis for 25 years now, and we have a very large cohort of patients. It's very well characterized, and much like Ellen, I believe that you can learn a lot by subsetting these patients by their autoantibodies, prognostically, response to treatment, lots of different things. Just to take off on that part of it, I've had a very long-standing interest in interstitial lung disease, and with Dana Asherman, who has since moved on to Miami. Dana and I have had a lot of collaborations on this devastating complication. I get more calls, emails, have 2 pending that I've got over the past few days on what to do with interstitial lung disease. It unfortunately kills my patients, and it keeps me up at night when I get these calls and these emails on the devastating complication of interstitial lung disease. So we've had a long interest in that, looking at outcomes, looking at predictive factors, and looking at treatment for that problem. And then finally, we're participating in a couple trials. One trial is an Acthar gel trial that we're doing. It's a smaller open study just to get a better feel for patients that are treated with this particular medication, and to see if we can identify anything more in terms of how they respond by collecting samples on these patients and trying to determine what types of studies to do, not only in looking at the response to that medication, but also looking at the patient repository, the serum, to see if we can see if there's any kind of biomarker there that is helpful to us. And then finally, we're going to be involved in another clinical trial. I was involved in a large trial of rituximab in myositis a couple of years ago that has been published, and we're still looking at data from that as well, learning from that. But this new trial is looking at another biologic agent called tocilizumab, which targets a substance called a cytokine, interleukin-6, and we're looking at a smaller trial participating with a total of 7 other centers, and we'll be beginning that trial in the next couple of months. Again, half the patients getting drug and half the patients getting placebo, to see if we can learn if there's another potential target for treating myositis patients.
Good morning. My name is Tessin Mozaffar. I'm a neuromuscular neurologist at University of California, Irvine. I trained with Dr. Pestronk in St. Louis back in the mid-90s, and at that time developed an interest in autoantibody-associated myositis, mainly from the perspective of whether we can predict a clinical picture based on the antibodies, but also see if there are differences in terms of muscle pathology. I since have moved to University of California and have built up a fairly large practice. We have a joint rheumatology neurology clinic looking at myositis patients. We take care of a large number of myositis patients, especially inclusion body myositis. And one of my more recent interests is looking at the new antibody in inclusion body myositis that Dr. Greenberg described, but looking at in terms of whether the presence of antibody predicts a different disease pattern in patients who are positive for the antibody versus negative for the antibody. We are also very heavily involved in clinical trials and participate in most of the major clinical trials that industry or investigator initiated.
My name is Dana Asherman, and as Dr. Otis said, I'm currently at the University of Miami, a rheumatologist. And echoing what I think is a pretty common theme here today, I can certainly remember my first patient with myositis who was kind enough to guide me and follow me into fellowship, where I continued to care for her for a number of years. I think more generally, my interest in this disease stems from an interest in how the immune system, the many ways in which the immune system interacts with the body, both in health and in disease. And obviously there are a lot of intriguing questions, frustrating questions in myositis? What's the connection between muscle and lung, for example? And I think it's these questions that have really fueled my current research interests. On the one hand, I spend a lot of my time looking at basic disease models in mice, actually, trying to figure out mechanisms of disease. At the same time, though, I have a translational interest in looking for blood markers that can help us identify patients who maybe are more at risk of lung disease, which, as Dr. Otis and others have said, is unfortunately a frequent complicating factor in these diseases. And outside of the research arena, this interest in the connection between muscle and lung disease has led me, along with one of my pulmonary colleagues, to develop an interdisciplinary clinic at the University of Miami, where we not only discuss the patients together, but we're in the room with the patients at the same time. And I think that that's something that seems to be appreciated by our patients, to know that they've got people looking at the problem from several different angles. So hopefully between these collective clinical and research efforts, along with my colleagues, we can continue to make progress and figure out better ways of treating the disease.
Good morning. I'm Lisa Christopher Stein. I am currently at the Johns Hopkins Myositis Center. Andy Mammon introduced our collaboration. We still continue to collaborate. I'm a rheumatologist by trade, and I became interested in myositis, much like many of these folks up here who I'm privileged to collaborate with, was my first patient who's in this room. She'll introduce herself to you. She's very proud of it. And I'm proud that I think she's been at every single myositis conference. I will say that I learn the most from my patients, and because you are a teacher to me, I spend my life work in clinical research. I don't go to the lab, but I'm fortunate to participate in what's called translational research, which brings things from the bedside to the lab bench and then back again. And my interest, as I'm reflecting back, I've been on the board for 5 years. I'll rotate off this year. And thinking back of how my interest began, in addition to that first patient, was a mentor by the name of Paul Plotz, who is still very involved in the work of myositis. And Dr. Plotz was at the National Institutes of Health. And I was thinking back when I first went to these meetings, which was back in 2001, 2002, discussing IBM. I was embarrassed to say that honestly I thought initially we were talking about a computer. I had no idea that what this disease was initially at that point as a rheumatologist. And interdisciplinary work has led to my work today. So to highlight what I do, in addition to helping to run a large cohort of patients, we've had over 1,600 patients referred to us. Approximately about half or a little more have a bona fide diagnosis that's verified of an autoimmune myopathy. And I cast my net wide. I have a lot of interest in highlighting a few of them, in addition to pharma-sponsored clinical trials, which we will participate in this year in 3 of them, including a BAF312, which is a novel agent that looks at trying to keep lymphocytes, the bad actors at the muscle level, in their lymphoid organs rather than going to the muscle cell. The hope is to prevent the egress out. And we're about to embark on a study looking at, with IDERA, who is here, Pharmaceuticals, looking at the effect of inhibiting something called a toll-like receptor. These are very novel ways of trying to prevent damage at the muscle level. Clinically, I'm also very interested in calcinosis. It has been very difficult, and I don't have the magic answer of how you treat it. We have just been funded to have novel imaging look with very sophisticated PET scanning to look at the extent and burden of calcinosis in adult dermatomyositis patients, which in our initial research has revealed that up to 38% of patients who self-report calcinosis as adults. I'm interested in cardiac complications, and I'm fortunate to collaborate with great cardiologists at Johns Hopkins. It's always been, obviously, the heart is a muscle. It's different than skeletal muscle, but there's some early indications that there may be some cardiovascular disease. Along with that, I have interest in statin toxicity and the safety of statins in myositis. Excuse me. And finally, I spend time fortunate to collaborate with Dr. Eliane Alexandersson from Sweden, and we've been doing focus groups, one of which we'll conduct here. And the reason that we began embarking on this initiative is because there are no specific patient-reported outcomes in dermato and polymyositis. And a patient-reported outcome is something that you derive. And what I've learned in doing, had no experience before, is that patients can actually tell us what's important to you in the context of both your clinical care and a clinical trial. And I'll highlight just one observation. I've always said that the patient is the best teacher. And what I have learned, along with my colleagues together, you've taught me that as I've learned that this disease is a painless disease. In medical school, that's the first thing I was taught, it was painless weakness. But focus groups in South Korea, in Sweden, and throughout the United States have taught us that several of you, arguably a majority of patients, have at one point reported muscle pain. That's just one small thing that we ignored before, but you continue to teach us. So I thank you.
Good morning. My name is Mazen Damashki. I'm a neurologist at the University of Kansas Medical Center. This is my first time being on the Medical Advisory Board. I've been recently appointed, and I'm really refreshed to see the intense participation by you, the patients, the caregivers at this national meeting. But what I also see are challenges and opportunities. I'm seeing scooters, and it tells me how much of an opportunity we have to make an impact on your disease. My interest in myositis started 20 years ago when I was in training, continued throughout the years. I've worked on HIV and myositis. I've worked on areas of gene expression profiling in myositis. I characterize myself that what I do currently is being not only a clinician educator to physicians and patients alike, but also a clinical trialist. So we currently are involved in studies that try to understand better the cause of the myositis. For example, in collaboration with Dr. Fred Miller at the National Institute of Environmental Health, we're looking at SNP profiling, single nucleotide polymorphism in patients with dermatomyositis. In collaboration with folks in the UK, we are looking at IBM gene expression to determine why some patients get IBM and normal folks don't get that. We're also involved in treatment studies. Most of you have heard of the IBM study of Novartis. We're one of the participating sites for that. We're involved in the other two Novartis studies looking at the BAF312 that Dr. Christopher Stein discussed earlier, a drug that traps lymphocytes inside lymph nodes in both polymyositis and dermatomyositis. And then we're delighted to be one of the participating sites in the future study that's just about the start of Dr. Otis, looking at this molecule, tocilizumab, that interferes with interleukin-6 in patients with PM and DM. But our own interest has been in a pilot study that was a collaborative effort between our group and the UK group in studying a chaperone protein that is called arimoclomol. And we did a pilot study in IBM patients that showed the drug to be safe. And next month, we're going in one more time to the FDA for a submission of a grant to seek funding so that we can conduct this clinical trials in people with IBM. One last interest I have is there are so many treatment approaches, different doses of steroids or adjuvant drugs, which is the best treatment. So we're hoping to launch into a study that looks at phenotypes and treatments that predict better response in people with myositis.
Good morning. My name is Jiří Vencovský, and I'm a professor of medicine, and I work as a rheumatologist in the Institute of Rheumatology in Prague, in Czech Republic. This is in the middle of Europe. How I got interested in myositis, you know, this institution where I work, it's specialized to different rheumatic diseases, and there's always been groups of doctors interested in lupus, rheumatoid arthritis, scleroderma, and so on. But when I came there, there was no one interested in doing inflammatory myopathy. So after consultation with my boss then, we decided to set up a small clinic that would be taking care of patients with myositis, and I stayed with it for the last 25 years. We are interested. Initially, it was only me for many years, but then we grew up. So there are several colleagues working in the clinic and also in the labs that are interested in inflammatory myopathy. Of course, we are interested in treatment because it's very important. We don't have good treatment yet for the disease, so we organized several trials in the past with cyclosporine. Most recently, we had 2 investigators-initiated trials, one with methotrexate, the other one with abatacept. The first one was, we know the results, and it was a little bit disappointing for us. With abatacept, we will know the results in a few weeks. We also collaborate with pharmaceutical companies, with industry. We help them to develop protocols for the treatment. We participate in one of the studies that was mentioned by previous speakers, this BAF312 drug. We also now collaborate with another pharmaceutical company with the drug that's directed to interleukin-1 called gevokizumab. It's monoclonal antibody, but it's in the initial stages, so we don't know yet how it works. We also are interested in biomarkers that might describe the disease, may be associated with activity, may help us to predict the development of the disease. So one of my colleagues is interested in BAF. This is a cytokine that stimulates B lymphocytes, cells that are probably implicated in the disease. She found that associated with the activity, associated with the level of autoantibodies, and she's now trying to find out whether there is some genetic predisposition to produce higher levels of BAF. And I have a colleague who uses ultrasound to find out how often is arthritis present in patients with myositis, and it was something also we overlooked in the past, that it seems that it's present almost in every second patient, and it might be the pain that we observe in this patient as well. And finally, coming from Europe, I think I need to mention that several years ago, together with Ingrid Grunberg and Hector Chinoy, we founded a network and registry in Europe. It's called Euromyositis Registry, and we put clinical and laboratory data into this registry. In many patients, these are longitudinal data. We have more than 3,000 patients in this registry, and it's a very useful tool to derive data for different studies, association studies, for instance, with autoantibodies. That's just being done now. Thank you.
My name is Stephen Greenberg. I'm a neurologist in Boston. I've worked on studying the causes and developing treatments for all forms of myositis. Currently, most of my focus is on trying to understand the cause of IBM, because I believe that understanding the cause of IBM is the best approach to choosing the best treatments and developing treatments for IBM.
Hi, my name is Ruth Ann Vlegels, and this is my first year here. I'm the dermatologist member of the panel. So most of my patients have dermatomyositis, and I feel very fortunate I get to see both adult and children with dermatomyositis. I work at Brigham and Women's Hospital in Boston with Dr. Greenberg, and also across the street at Boston Children's Hospital. My research in dermatomyositis focuses on the skin disease, so how the skin disease impacts patients and their overall quality of life, and then also thinking about treatments for the skin disease specifically. And I think one of my favorite jobs as a physician is teaching younger trainees and doctors about dermatomyositis. So I run the dermatology course at Harvard Medical School, and I make sure we always have a session designated to dermatomyositis, and some of my kind patients come in and help me teach the Harvard Medical School students about this disease. So hopefully we can get them interested from a young age in thinking about this disease.
Excuse me. My name's Todd Levine, and I guess my interest in myositis, like Tassine, goes back to when I trained with Alan Pestrunk at WashU as a neurology resident and a neurology fellow. My practice is now in Phoenix, Arizona. I'm also a clinical assistant professor at the University of Arizona. We do a lot of trials in our center. The two current trials that we're working on, one is a large registry of Acthar. So many of you have heard about that drug, and we're trying to get more data on patients that are being treated with it and what kind of response they have and whether they're a subgroup of patients based on pathology or biomarkers that might predict who's going to respond to Acthar. The other is a small investigator-initiated trial that we're doing in inclusion body myositis. This is with the drug called Tysabri, which is approved for MS. And as you've heard before from some of the other speakers, one of the ideas in myositis is trying to figure out ways to stop the immune system from getting into the muscle. And this is a monoclonal antibody, which seems very effective in stopping the immune cells from getting out of the bloodstream. And so we've been doing that trial for the last 6 months.
My name's Adam Huber. I'm a professor of pediatrics and a pediatric rheumatologist at Dalhousie University in Halifax, Canada. My collaborators and I had a number of recent publications looking at clinical subgroups of children with inflammatory myopathy, looking at antibody subgroups of children with inflammatory myopathy, and most recently we published on early predictors of mortality, again in children with inflammatory myopathies. Right now we're working on looking at predictors of calcinosis, which while an important problem in adults, is a tremendous problem in pediatrics. And the last thing we're working on right now is looking at non-clinical trial approaches to studying treatment, because given the rarity of illness, it's extremely difficult to conduct clinical trials in pediatric myositis.
Well, thank you all for your introductions and your contributions to the field. I mean, I'm sitting listening to this. I'm very excited about what's going on today. I think it's really incredible the amount of work that's going on that's improving our understanding of these diseases. I think several people made the point that the terms polymyositis, dermatomyositis, and inclusion body myositis, although names for diseases which many of you know you have, are really inadequate to help us understand what therapies to use. And there's a lot of work going on to find out the subsets and the predictors of what disease manifestations might occur and what treatments might be best used. And that's working at one level, finding the cause, finding predictors that tell us what to use. But on the other hand, we heard about 6 different clinical trials that are ongoing at this time or will be started soon, trying to find better medicines, better treatments for patients. And the first public statements I've heard about the trial of injecting the follistatin gene into muscle, as far as it's having a positive outcome for children with Becker's muscular dystrophy and in the patients who got high doses with inclusion body myositis, that's incredibly exciting. And a lot of this work has been sponsored or facilitated by TMA. The other thing that's important to recognize from the panel is that the study and treatment of any form of myositis is really a multidisciplinary activity and is represented by this board. We have neurologists, rheumatologists, pediatricians, dermatologists, physical therapists. This board represents that multidisciplinary approach, and TMA brings this group of people together. And there's no other venue where this group of people meet. And I had the pleasure of listening yesterday to 4.5 hours of scientific presentations that these individuals gave to each other, sharing not what's been published, but what they're working on, what their ideas are. So this has been an incredibly important activity that TMA has made it valuable. And I want to again thank all these members of the Medical Advisory Board for their service to the TMA, but also what they're doing for understanding and making better treatments available for patients and what they do for them. And I think they need another round of applause. So now you've heard what they do and what their interests are, and perhaps they have stimulated some questions from some of you, and we'd be happy to entertain them. And we'll start with Augie, first one.
Thank you. First of all, I'd like to add our thanks from the Board of Directors and from all the members of the Myositis Association for your service to the Medical Advisory Board. It is well appreciated. Now, I understand that everybody up there understands fully the negative effects that myositis has on a person's life. And despite all of the research that is ongoing and all the hope that is out there on the horizon, we have many members now who, especially who are in advanced stages of inclusion body myositis, that are looking for answers for something that could help them today. It's one thing to know that we're 5-10 years from now, there might be some treatments or maybe possibly even a cure. But the folks that are in advanced stages that are just losing quality of life are looking for answers. And recently something has come up that has got the attention of many of our members, and it's got to do with a stem cell therapy, whereas the stem cells are harvested from fat and then re-injected, unmodified, back either directly into the muscle tissue or infused into the body. Now, we've got reports from many of our members who have already had this treatment and reporting positive results. Now, these are unverified. We don't know if they're actual results or if they're just imagined or a placebo effect. We just don't know. However, when somebody is in a desperate need for a solution, for a remedy, they're going to go out and they're going to be willing to risk and try something that I know all of you are going to consider experimental. So I was hoping that you might be able to address to our members, give us some guidance on this, even though you may not be fully aware of the specific procedure. I was wondering if you could address this issue of stem cell therapy, especially for IBM.
Dr. Mosafar, you want to take first crack at that one?
Sure. Bob Goldberg initially had asked me to respond to this when this news first came out, and partly also because not only in my status as the chair of the Medical Advisory Board, but also that this particular stem cell group was in my backyard in Irvine, California, not associated in any way with the University of California, Irvine, which in itself is a very active stem cell research center. We've seen this phenomena before. We've seen this phenomena in ALS, which is in some ways a much more aggressive disease and fatal in majority of the cases. And we've seen this phenomena in other diseases. As your medical advisory board, our job is to give advice on whether this is a medically sound treatment, whether there is solid research behind this treatment, and if the risks that you're taking is worth given the potential for benefits in there. And based on this, the Medical Advisory Board, with its experience and with the review of the literature, had come up with a statement saying that we do not find that the research behind this particular form of stem cell treatment is validated. There's actually very little, if any, research behind it. The risks are definitely there. We are talking about multipotent cells which can transform into a lot of other unwanted structures. So therefore, there are risks of cancer, there are risks of other deformities that can happen. And I personally had an objection to advertising this particular treatment as a cure for a myriad of diseases that are not even interlinked and charging money for it. If it's truly, it needs to be done as a research study, which means that by definition, research studies usually do not charge patients for treatment, and there needs to be very close scrutiny of any treatment-related side effects, any treatment-related benefits. What we are seeing is a bunch of anecdotal reports which are not verified or validated. Okay. I don't think the medical advisory board in any way is saying that this treatment potentially cannot work. It just needs to be studied in a controlled scientific fashion with close attention paid to treatment. But we would not at this point, given the lack of scientific evidence behind it, we would not routinely recommend this as a potential treatment, despite the fact we very well recognize that especially IBM, where we don't have any treatment, is a desperate disease. And some of you are in stages of the disease where you may not be eligible for clinical trials, but at the same time, we don't feel that the risks are worth given the very small benefit potential here.
Would anybody else like to comment on the panel? Okay, any other questions?
It's on.
It's on.
Here. Here. Okay. Oh. So I was sent an article yesterday by my dad, who's always concerned about my health, out of a magazine called Science Daily. And the headline was, "Scientists discover how to switch off autoimmune diseases using anti-antigen therapy," or something like that. This came out September 3rd. It's from Bristol University. Is there any validity to this? Anything that you folks are doing that are similar? Have you heard of it? Okay, that's my question.
Well, yeah, I mean, it was just a kind of a news flash, but I mean, I don't know a whole lot about it, but I read the article, and it's basically it's an approach to shut down very early an autoimmune disease process. And if it's relevant, it's of course going to catch the attention of a lot of people interested in autoimmune research. All I can tell you from what I read, and maybe others read more on it, is that it's interesting, it's promising, and something that kind of addresses such a proximal step, an early step in autoimmunity is naturally going to catch the attention of the news wires. And my opinion on that is that we have to see where this goes, and it really has to be published in a peer-reviewed type fashion before you can go further.
For those who have questions, just put your hand up. We'll bring the microphone to you.
Hi. First of all, I'd like to say that it's great to see how the board has expanded in the number of the variety of professionals. So that's great news for our group. My question has to do, I think it's a piggyback off of Augie's question. Is there anything being done with stem cell research that may prove hopeful for those of us who have lost muscle and are looking to, in addition to exercise, get back some muscle mass in the near future, in the not too distant future?
I can't see who's asking the question. Can you? Oh, thanks. So I'm going to speak about one thing that's non-stem cell related, but it's related to the previous procedure to stem cells. So I'll give you, while non-published, but hopefully soon to be, I'm going to give you a small glimmer. We have not looked exclusively, we haven't looked at all in inclusion body myositis, to be clear. But in some of the more severe myopathies, one of which is associated with signal recognition particle, or SRP, that tends to be so severe it can look like a muscular dystrophy, especially if you happen to be African American in our cohort. There was a fairly talented hematologist by the name of Rod Brodsky, who at Johns Hopkins initially realized that it wasn't the stem cell transplant. This we're talking about a marrow transplant for somebody who has something like, there was a disease called aplastic anemia, which sounds benign, but it essentially means that your entire bone marrow is wiped out, and previously required a bone marrow transplant. What he recognized was it wasn't the transplant, but it was what's called ablative therapy, or ablating, or taking down the bone marrow essentially to zero, using high doses of cyclophosphamide or Cytoxan. Many of you may have been exposed to Cytoxan in smaller doses. But the earlier therapy looked as if just ameliorating the bone marrow itself, taking that down without ever having to transplant the patient's own cells, allowed the bone marrow essentially to reboot. And so I'll tell you that I have anecdotal and hopefully soon fairly scientific evidence that that procedure, while expensive, actually has been paid for by some insurances. So high-dose cyclophosphamide may be a nice adjunct to not having to do a true transplant or a bone marrow transplant. This is a little different than the stem cell transplants that were talked about. So it's one option in the future. We really have not looked at it in inclusion body myositis, although it's not without merit to maybe potentially consider it. So there may be less toxic and unique therapies that may matter in the future. So I'll just give you a little glimmer.
I'll make a brief comment as well. I'm certainly not a stem cell expert, but I think for those of you who don't know this, when we use the word stem cell, we're talking actually about a lot of different cell types. And so there's hematopoietic stem cell transplants, which are sort of blood-derived, something called mesenchymal stem cell transplants. And I think it's the latter really that is being looked at mostly in terms of regenerative potential, not only in diseases like IBM, but other neurodegenerative and really a myriad of conditions. And I think that the short answer to a very complicated question is that this is an area of intense research, not just in the field of muscle disease. And some of it's very credible, but it's just at an early stage, and it's not ready for prime time yet. But I think that there's hope even in the medical community that over the next 5 to 10 years, as we learn more, that it could have potential therapeutic benefit.
Just to echo Dana's point, the neurologists up here deal with a lot of. Can you hear? Yeah. So the neurologists up here, we deal with a lot of untreatable, progressive, fatal, disabling diseases. And so we're asked these questions about stem cells daily or weekly. And again, I think Dana's points are exactly what I would say. I just wanted to echo them so you've heard them before. We all believe stem cell treatment will have a place in medicine, hopefully in all of our lives, but certainly in some of our lives. But we don't believe that what people are doing now is effective. And so it's really with 100% certainty we say that it's not something that's worth spending your money on, and at worst, it's risky. But we believe that it's going to have a place in medicine and in some diseases and will be groundbreaking, but it's just not there yet.
Yeah, my name is Tom Stone, and I disagree that the risk-reward issue, that it's more risky than the reward at this point. At now, all these clinical trials, if they're even successful, in which they may not be, the medication won't be out for maybe 2 to 5 years. There are a lot of us who our muscles are atrophying at a high rate, and it may not be even usable at that point. So at this point, stem cell treatment, which to my knowledge, the stem cell treatment that the people who were on the Myositis website who have done it, have done their research as best as possible, and have had high success. I can name one example. The gentleman he was at the point where he couldn't roll over in bed. After he had the stem cells, he now can roll over in bed by himself. He couldn't raise his hands above his knees when he's on his wheelchair. Now he can raise his hands above his head. He couldn't put his feet on his wheelchair supports prior to the stem cell research. And within months after that, he can now put his feet on his wheelchair by himself. And he moves his hands much better than before. So my point is, there are a number of cases like this out there. There's a case it was on the news media recently where a woman was going blind in California, and they were putting on heavy doses of prednisone, and she was getting all the bad effects from prednisone. After a while, the doctor, when her eyesight got down to the size of a quarter, they said, let's try the stem cell treatment. Within months, her eyesight returned to normal. She got off of prednisone and her life returned to normal. So stem cells do have a point, and we need to have someone seriously try to get this analyzed using clinical data rather than anecdotal data, so that we can have a treatment within 6 months or now, even though the risk is maybe $10,000 for the treatment, which a lot of us, I'm sure, are willing to pay if we have some positive benefit. And so I think the risk reward is there to at least take a real serious look at this.
If I may comment on what you suggested there, this is very resonant with me because as a neurologist, I've heard patients ask me that have a different disease, ALS, about going to China or to India to get stem cell transplant, and I've had to go through the literature and what's reported out there in the way of successes. For example, in some of the China experiments with stem cell in ALS, those reports of benefits were occurring within a few days after the transplant, which doesn't make any biological sense, as well as in areas where the transplant did not take place, which also doesn't make any sense. So while I cannot comment on the case that you are describing with all these benefits, I just want to caution everybody in the audience about, first, where were the stem cells injected? Did it make sense that muscles that were not injected would get better? Number one. And second, is there something called the placebo effect? And we have to be careful about that. But we know that what works right now for IBM, which is not a victory of medicine, let's really face it, it's a failure of medicine, but what works currently for IBM is a good diet, rich in protein, 30% or more, to slow down the disease progression, and physical therapy, exercises on a regular basis. And what these two do is not really have you gain function or strength back, but maintain what you have for a longer duration of time. But I compare IBM to ALS because it's just simply put, IBM is a slower form of ALS. So there's the good news, bad news, but then still, desperate case scenarios call sometimes for desperate ideas, but this has to still be tested. So as an advisory board member and new to this, hopefully you won't throw eggs at me or tomatoes at me, I would say be careful with these things. I worry about patients being taken advantage of because these are expensive procedures, several $1,000s, $10, $15, $20,000, and out-of-pocket expense, insurance won't pay for it. So that's putting the medical unknown risks aside, which I'm not sure if they're there or not. I worry about protecting my patients from unproven therapies. That's where I stand at this.
So we could probably have a 4 or 5 hour discussion on this particular topic. And I'd like to switch away from it with the final statement that we all understand, everybody in this room understands how people who have a condition that is not treatable are desperate and want to look for anything that they think might help and are willing to try it. And nobody here would say you have a right to do whatever you think is right for you as a person. You're not saying don't do it. We're saying we don't recommend it because there have not been any trials performed where we know the patients who are in the trials have the diseases and the outcomes are validated based on specific measures that are tested. That data doesn't exist. We wish it did. So I hope that can be enough on that particular topic today. At the formal session afterwards, I'm sure any of us would be happy to talk to you individually about it.
Hi, my name is Melissa and I'm here with my Aunt Joyce. I have polymyositis and she has IBM. And so we were interested in the comment from the physician that's looking at cause in terms of treatment. And we wondered if there was anyone looking at DNA in terms of any sort of familial predisposition. And also I'm interested if there's been any look at DNA in terms of changes to DNA that have been made through exposure to environmental toxins like Agent Orange, which has also come down to me through my father who served in Vietnam.
Steve, you want to take first track of that?
There are a number of people who are looking at DNA sequences in patients without a relative who has a form of myositis, as well as small family groups in which there are myositis in more than one member. I know none of this has been published yet. There's a large group in the United Kingdom that's sequencing hundreds of patients with IBM. They haven't published their data. There's an investigator at Washington University who's done some sequencing. That hasn't been published. I've done a fair amount of this myself and have not published any of the data. I have not found a genetic variant that I think is a cause in related individuals in blood DNA. I'm still looking though at other populations of DNA. Your other question is about whether there can be non-sequence changes in DNA called epigenetic changes in DNA. And typical ones that can be looked at are methylation, for example. And I think that is a potentially productive area of research. I'm not aware of any researcher who's looked at all at epigenetic changes in DNA, like what you suggest with regard to Agent Orange, potentially, in any patients with any form of myositis. But I think that would be a productive area of research.
I can just add that I think that's a great question, and one that everybody's interested in. And there will be, I'm pretty confident that I could say that there will be research results that come out probably in the next 5 years from just the same groups that Steve has talked about, and even groups. I received an email today on a major effort that people are going to have looking at the correlation of specific HLA sequences, which are chromosome sequences, in different subsets of myositis. So without being more specific, I can just tell you that there's a lot of people that are working on that.
This question is probably directed toward Dr. Todd Levine. It's in regards to ACTAR. I understand that you're trying to get other physicians interested in prescribing this so that they can then become part of your data. My question has to do with before, my understanding is this was available way over 50 years ago. And I was told it was also used for PM and DM back then, but it fell into disuse or it wasn't advocated. And now through your efforts, it is being advocated. What I'm concerned about is the price that it was 50 years ago, and now all of a sudden it's at least 1,000 times more than it was then. And I know one physician can't justify that, feels it's unethical to have that much of a price increase. And also, since I've been here, I've talked to patients who have had some of it, but was being charged so much and can't afford it, and so is no longer taking it. So I guess I want you to speak to that, those issues, and how can someone be on it with that particular price, even though he was rejected from his insurance company, talked to the company, they did not offer him assistance. And yet when I talked to the company yesterday, they were touting they gave $400 million in assistance. So it seems very unclear.
So that's a lot of questions. But briefly, so the history is the drug was developed as a way to have your body produce steroids before people had the ability to synthetically make steroids. So that goes back to the 1950s. So the drug has been around for 60 or 70 years. Over the last 20 years, it's become clear that there are probably other mechanisms of action for the drug other than just producing steroids. That there's a whole other pathway of the immune system called the melanocortin receptor system, and ACTHAR and ACTH probably act at that system and may have other effects other than just the steroids. I would say that my interest has been not so much in trying to convince other doctors to prescribe the drug, but since the drug has the FDA indication, there has not been any attempt to collect the data on the patients that are treated. So if physicians are going to prescribe it, my interest has been in to say, as you've heard up here, we want to collect information on patients. The effort in Europe to generate a registry, registries are not really controlled trials, but they allow us to learn a lot in a much cheaper way about large numbers of patients. And so my interest has been in just trying to collect that information. The cost issue is a very complex issue, and I think all of us as physicians and as patients and politicians have to recognize the fact that there's only so many dollars that the healthcare system has, and certainly there's been an effort to try to treat patients in not only an evidence-based way, but also in comparing effectiveness. And so if you've got two drugs and one drug is $100 a year and one drug is $100,000 a year and they're equally effective, then obviously the drug that's $100 is the drug that we should be using. So I think the positioning of Acthar, because of its expense, is clearly behind other drugs such as steroids, which are dirt cheap and very effective for poly and dermatomyositis. Having said that, many patients don't respond to steroids. And at that point, we talk about other drugs. And the example that I use is IVIG, which many patients have probably been on in this room, and also costs hundreds of thousands of dollars a year. And so we do use very expensive drugs to treat these diseases. We have to be cost effective in terms of where we position them so that we don't start with a drug like IVIG or a drug like Acthar if somebody responds to corticosteroids. But if somebody has failed multiple treatments, then I think if we have a drug that may potentially work, and in the position of Acthar where it actually has an FDA indication, I think it's reasonable to consider using it. I will, QuestCore has sponsored my study, and so as a disclaimer, but they have been, at least in my patients, usually very helpful in terms of helping with patient costs. There are certain restrictions all companies have to adhere to from a regulatory standpoint, but in general, I've had several patients that the company has just given the drug to through a system called NORD, and which allows drug companies to give drug for free or to help with copays. So I would say the company has been helpful, but they do have rules they have to stick to from a federal standpoint.
Wow, I am so impressed after being around the myositis community for over 20 years to see the quality of expertise, the passion of the panel, and the variety of things that are going on now. When my husband was first diagnosed, one of the things that was told to him is, gee, I wish I could tell you of something in the works, in the pipeline, something that might be promising, but there isn't anything that I'm aware of. But I have a question, because as I listen to all of you, and I know some have had the opportunity to meet some of you in the past, you all referred to that you've been in the business, if you would put it that way, for maybe 20 years because as a fellow, you became interested in it. Can you tell me how the medical community has grown, say to the point of where it is now? Are you seeing more and more fellows that are becoming interested? And maybe a little bit more, I know there's been a great effort by our organization and so forth for the medical schools, for the young interns coming up, to make them more aware of this specialty. So I'd like to know a little bit about what your thoughts are on the future of the experts. Thank you.
I don't see any nods or reaches for the microphone. So maybe I'll take that. I'm not on the board, but I'm on the board, but not on this one. But I was for a long time, and I was involved in medical education throughout my career, including chairing 3 different departments of internal medicine. The way, we have students, we have residents, and we have fellows, the 3 levels of training. I'll get exposed to the topic of myositis probably at each level, depending on where they are and whether there's somebody in that institution that's interested in myositis, their exposure can vary to minimal to quite a bit. One of the present activities that was sponsored by QuestCorps is to reach out to medical schools to provide them with a lecture for a grand rounds or some other conference to their medical students or to residents or fellows or a combination of them. All 125 medical schools were reached out to, and 32 responded that they would like to have somebody come and speak to them about myositis. We've arranged for about 20 of those visits and are actively working on getting the remainder of them satisfied with a date and a person from the board who will come and speak to them. Unfortunately, there are just not as many people interested in myositis as we would probably like, but I know every one of us who is, when we have an opportunity to work with trainees, we're passionate about sharing that information, as Ruth Ann said in particular, and hopefully this is effective. Another thing that will be more effective and get people more interested as we learn more about these diseases, and there are more treatments available, and that is what is going on right now.
Hi, am I on here? Okay. My question is about medications which are not anti-inflammatories, and it's a two-parter. When I was diagnosed with my inclusion body myositis, my practitioner put me on a cocktail of over-the-counter medications that included L-carnitine, coenzyme Q10, vitamin B1, vitamin B2, did not put me on any creatine. Okay, that's one part of the question. I came to her having been on statin, and last year the question, do statins cause inclusion body myositis, was definitively answered as no. But is there any reluctance on any of the practitioners to maintaining someone on a statin with myositis?
Christina, take that one.
Absolutely. I'll let someone else take the first part of the question. I'm not sure what you're asking, or we'll come back to that. I have several patients, and because we are certainly interested in cardiovascular disease, and there is an increased risk of heart attack and stroke in patients with myositis, we're looking at cholesterol levels, and we're looking quite frequently. And I have several patients with myositis who do take their statin. I think certainly it's reasonable when you first meet a patient with complaints of muscle weakness that the statin is pulled off, and that happens quite frequently because you don't know what the diagnosis is. And as Andy and Chris and Lisa have clearly shown, there is a myopathy associated with statin use. That being said, once you know what's going on, you have to treat the patient, and treatment of the patient involves other things besides the muscle disease. And one of those is cardiovascular risk, which just not in myositis, but in all of our inflammatory diseases and many of the autoimmune diseases that are non-inflammatory, there is an increased risk of cardiovascular disease. And so I have several patients who are back on their statin and doing quite well. Certainly the best scenario is if you can control the myositis and then gently reintroduce the statin at a lower dose initially. The statins can have myopathic effects, myalgias, that sort of thing. If the patient has not had that in the past on their statin, it's less likely that they're going to get it as a new symptom. I'll let somebody else answer the question about the vitamins and the creatine. I don't have too much history in using things like that.
I was going to take a stab at that one, if that's okay. So I've actually had an interest in supplements for a long time, and in another disease where patients get nerve disease called peripheral neuropathy. I've taught a course for several years at the American Academy of Neurology on that. So the bottom line is, I think as Tassine mentioned, probably the best thing is a good diet and exercise. The problem is we don't all eat a good diet, and so then we start to take vitamins. There's probably no evidence that taking extra or mega doses of any vitamins is really helpful, But as part of a healthy diet, taking some supplements probably does make sense. Creatine's been studied in a number of different muscle diseases and never found to be helpful. Again, it's just giving your body the protein that it needs. When we talk about risk and benefit and patients ask me those questions, it's the same issue, which is if you ate a really well-balanced, healthy diet, would you need to take extra vitamins and supplements? Probably not. Do megadoses of any vitamins and supplements help? Probably not. Do vitamins and supplements help to form the basis of a well-balanced diet and an exercise program? Yes. And so it may be worth it, but we don't have any data that it works.
I'll add one other comment. I do have several patients who have their different supplements that they like and feel are of benefit. And while I may not have data on the specific supplement, if it is something that is rather harmless, like a vitamin B, and the patient feels that they are benefiting from that, I do not discourage the use. I just can't say about data suggesting particular supplements.
Can I add a couple comments? This is Renee Lantner. I'm on the board of directors, and some of you have seen me giving the nutrition talk. So, Dr. Levine, I just wanted to ask a question because we've got Dr. Lundberg's colleague here, and in researching stuff for the nutrition talk, my understanding was that there was a Cochrane review that looked at her data in 2007 of the creatine, and it was felt to be helpful only for DM and PM, not for IBM. And so I wanted to make sure that there wasn't some confusion there, because my understanding it was helpful. In my research also, L-carnitine and coenzyme Q10 were found not to be helpful for any of the inflammatory myopathies. But I wondered if you could comment on the creatine, because it was my understanding that it is helpful.
That's true. There is one randomized control trial where we tested the combination of creatine supplements and exercise and a placebo and exercise. And it turned out that the patients who got the creatine supplements in combination with exercise improved a little bit more than those who only exercised and got a placebo. And that was in established polydermatomyositis. There are no studies in inclusion myositis, although I have a few patients at the Karolinska who do take, with IBM, who do take creatine supplements and who do feel some benefits in terms of maintenance of muscle function or maybe improvement. But I want to underline that you have to exercise in combination with the creatine.
It's a really interesting question. So again, I've been interested in the supplements for a long time. And actually, not in myositis, but I went to the FDA to get a trial to study it. Somebody asked why not, which I thought was a great question in the audience. This is the answer to the why not question. And so the difficulty is that these supplements have no regulatory control to them. And so when I went to the FDA and I said, I want to do a study of supplements, the first thing they said is, you're going to have to prove to us what these things are. So you need to do stability analysis and composition analysis, and it was actually over $1 million to be able to verify to the FDA what's actually in the supplements. And so the difficulty in terms of doing studies with these supplements is not so much that the studies are that hard, it's that the supplements are so poorly controlled. And so there's another whole layer of difficulty with this. So I'd love to do studies with these things, and in particular combinations of supplements, because I think the point being that the benefit we see from taking any one supplement, whether it's creatine or B12 or acetyl-L-carnitine, is probably very small. But then the question becomes, well, can you do a combination where you took multiple supplements because they're relatively cheap and easy? And the answer is the FDA makes it almost impossible to do that.
So I was going to add one more comment to this. So a lot of our patients take multiple supplements, and not only does it add to the cost of all the supplements, because none of these are covered by insurance and you have to pay out of pocket for that. But the other concern is the potential interaction with some of the medications that you may be taking. We know very little about these supplements, and as Todd mentioned, we don't even know how well they're regulated. But more importantly, we don't know how they interact with some of the essential medications that you're taking. So unless there is a clear benefit I mean, again, I have no problems with somebody taking a tablet of multivitamin on a daily basis, but some of these supplements may potentially be dangerous on that. So again, unless there is evidence that supports their use, I would be cautious in general.
Well, there is some rationale. It takes a leap for the use of carnitine, creatine, coenzyme Q10, vitamin C, the supplements that Dr. Lawesser mentioned. And it comes from studies using these agents in a rare group of conditions called mitochondrial myopathies. And these are diseases in which the cells of the body have difficulty making energy. And so all of these supplements, it's felt they have a role in energy production. And by supplementing them, that will help. And it's been shown in mitochondrial myopathies that it is somewhat beneficial. Also, there are people who are deficient in coenzyme Q10, and giving them coenzyme Q10 helps. So it's here's some supplements that can help people with very rare conditions that do affect muscle. Would they also help people with other conditions that affect muscle? And we really don't have much data there.
We have
Move
another
on to
question
another
back here. Oh,
I have. I wanted to know, first of all, how do the people, the doctors, be admitted to these studies? I'm from Salt Lake. I go to the University of Utah, seeing a neurologist who said that he was disappointed that he wasn't involved in one of the studies, that he himself would be the one that would be managing it for Utah. How are they selected? I don't have a clue. And then, I guess that's fine. Please.
Well, I'm going to give this one to you, Chet, since you organized the biggest trial in myositis in the history. So.
Thank you, Bob. So I had a response too where we weren't invited for a particular trial, so I can understand that doctor's response. But I mean, the answer to that is probably complicated because it depends who initiated. To get into a long answer, let's just try to make it simple. So there are some trials that are what are called investigator initiated. So it is up to the investigator, such as if I'm doing a trial now and I have to determine the sites that I think are going to be able to enroll the patients, and perhaps the sites that have had experience in the same collection of data for the trial that I'm doing. So when I did the very large study in rituximab that Dr. Wortman is talking about, I went to the major myositis centers, or the people that were used to the measures in that particular trial. And that's who I went to. And I included neurologists and rheumatologists, and there were even some dermatologists peripherally involved. So that was an NIH-funded trial. Now, the investigator-initiated trial is one that's, like I just said, by the investigator, so they have to try to pick the site that they think is going to be able to enroll. Then the other one are the pharmaceutical trials. And the pharmaceutical trials that are sponsored, again, try to enroll and enlist the site principal investigators who have the most interest in that particular disease, and again, who are going to be able to enroll the patients that are necessary to meet the numbers sufficient to answer the question of the trial. So the simple answer to your question is that investigator, in order to be chosen for a particular trial, has to have a history of being a participant in a trial. If they don't have the history, then they have to establish the expertise, or they have to get on the Myositis Advisory Board or something like that in order to have the ability and the exposure to be chosen as a site. I don't know any other simpler way to answer the question. If anybody does.
Well, I mean, I think in the end, as Chet said, it really comes down to whether it's investigator-initiated or industry-sponsored studies. It really comes down to a lot of these trials have to be done rapidly. The recruitment has to be fast, which means that you want to work with a center which has a large number of these patients who have the research infrastructure, which means that they don't have to reinvent the wheel. So they have the coordinators, they have the evaluators in place. They have an environment at the institution which is conducive to clinical research. So that means the study is not going to get bogged down in the ethics board. It's not going to get bogged down in the contracts and research grant negotiation. So those are factors. So there are benchmarks by which trials sites are evaluated on that. It's not a perfect system, and a lot of the times it's chosen based on who you know and whether you have a profile, as Chet said, in a particular disease or not.
If I might add to this complex question, the following is that myositis being a rare disease, one of the issues with clinical trials that involve humans is that a lot of them stop. And you probably don't know why they stop prematurely. It's because of failure of enrolling subjects into the study. That occurs more than half the time. So when I propose sites for my studies and I contact people, I like to know that A, they have the patients, B, they have the staff, the facilities, the resources to be effective in helping move science forward. And having a track record of having done it before is extremely helpful. It shows that, yes, not only can you talk the talk, you can walk the walk. So simply put, a lot of things go into this. But studies still in rare diseases, especially with enrolling people. I can give you an example of a different rare disease. We ran a study on myasthenia gravis, another rare disease that weakens your muscles. We started off with 10 sites. We thought we could get 50 patients with 10 sites. We went up to 14 sites, 18 sites, 21 sites to get those 50 patients. And this is a year and 2 years into the course of the study. So it's always challenging to do enrollment and recruitment in rare diseases, and certainly myositis is a case in point.
Mike?
Hello? Okay. Am I coming through now? Okay. If I may direct my question to Dr. Greenberg, I think you mentioned that you are working on the causes of the illness, I think IBM. And I wanted to know if you'd be able to share any of that information with us today, any of your findings.
The answer is no to that. I've certainly published a lot on the mechanisms of this. So there's different aspects to understanding the cause of a disease. What perhaps is more important is the mechanisms of it, what's sustaining the muscle injury and the disease. It certainly would be nice to find one thing that set the process off in the first place, but the really key issues are what is sustaining the disease, what's causing the muscle injury now in people who already have the disease. And certainly there's a large aspect of the immune system involved in that. And that's the only aspect I'm sure about, that there's immune injury to muscle. I'm not sure if there's any other mechanism that's causing muscle damage. So I am working on what aspects of the immune system in particular are involved in the muscle injury and trying to narrow that down. Along the way, I led a team that discovered a protein in the blood, an antibody, which I don't know if it's causing damage or not, but it's useful as a blood test for IBM. I think that's been mentioned. Dr. Mosavar presented some research that he's done in that area. So I am working on it. I do have ideas that I'm not fully sharing with everybody right now, but they are along this line of the nature of the immune reaction in injuring muscle in IBM. I hope that's sufficient.
Well, it is. It at least gives some idea of what's going on. I was actually diagnosed with polymyositis like 8 years ago, and what I'm finding is that the treatment, either the medicines aren't working, or if they do, but they're severe side effects. So, and now the illness is starting to act like IBM. So I'm trying to find out what exactly is going on so that I can develop a better treatment plan and get better.
Right. I think there's a lot of other people in a similar situation who are diagnosed as polymyositis initially, either because the person making the diagnosis isn't completely familiar with IBM, or because they do in fact look to every expert as if they have polymyositis, but then don't respond to anti-immune therapies and develop a picture that is more typical of what most experts will call IBM.
Okay, hi. I was wondering what you're seeing as the best or the most promising treatments for ILD. What you're seeing is working best or the most promising on ILD. I have anti-syntase, and I've had it for 4.5 years. I take Cellcept and Pred.
So I think it's obviously a very important question, and unfortunately another question where we really don't have the answer at this stage in terms of what's the best treatment for ILD. I think that you could ask several of us sitting up here and get several different answers, and I think that reflects the fact that it's extraordinarily difficult to systematically evaluate the effectiveness of treatments for, it's not a rare complication of myositis, but myositis is already a rare disease, and then we're talking about a subset of patients with myositis. So we can't do systematic trials at one center, and there's a lot of complications in terms of making sure that we're collecting data in the same way and evaluating outcomes in the same way. And it's a very active topic within rheumatology and pulmonary circles. Having said all of that, I think that many of us would say that we would start with agents like Cellcept. There's at least some anecdotal evidence, not so much in myositis as in other related autoimmune diseases that are complicated by interstitial lung disease. But to be honest, even that data is not superlative. I mean, it's in some cases the data is showing equal effectiveness to other therapies which are more toxic. So we almost by default arrive at Cellcept. And that's not to suggest that it doesn't work. I don't want to be too pessimistic. I think in many patients it does. There are certainly other agents. Dr. Otis has published several articles looking at tacrolimus, which is a drug like cyclosporin, that can work in particular in subsets of patients like you with the anti-synthetase syndrome. And then now there's all these questions about whether or not some of the so-called biologic agents, things like rituximab, might work for lung disease. And we just don't know the answer. There is many reports showing that it can make lung disease worse as there are saying that it can make lung disease better. And again, this is sort of a common theme in our answers to some of the other questions, but I don't think that we can fully address this until, as a community, we establish sort of what the right outcome measures are to look at, so that we can combine data from disparate centers. So I'm going to let somebody else add to that.
Thank you. I'm afraid we only have time for one more question in this format, but all these individuals will be around today. Many are giving lectures and sessions this afternoon, and I'll be willing to answer individual questions. So one last question.
Yes, it's really a two-part question. One is more probably a statement. I've seen 4 neurologists, and 3 of those really didn't have very much knowledge about IBM or myositis in general. And I'm just curious how you go about educating your fellow compatriots out there in that field, in that it's clearly not a level playing field. I think it depends on where you're located, whether you're in Cleveland or Atlanta or Miami or wherever. But some of them clearly didn't have much knowledge about IBM. My second question really is more of a question, that's for Dr. Sahenk, regarding the status of the follistatin trial and perhaps what are the next steps. that you anticipate going forward? Thank you.
I can probably try to answer first part of your question too. Major universities, institutions, Department of Neurology actually have established a fellowship program for neuromuscular disease. So those fellows who go through this training, I'm sure will become equipped with the knowledge to make the right diagnosis of IBM. In general, neurologists, they do go through some training, of course, about these disorders, but the time that they spend, may not be enough to be able to make diagnosis if they are on private practice and not constantly renewing their, knowledge about these disorders. The IBM trial right now, as I said, we completed the high-dose bilateral quadriceps injections on 3 patients, and we will go ahead and recruit another 3 patients, so that will be a total of 6. But the idea is obviously the more we do, the more we will come up with convincing data that it is working. So far, 3 out of 3 in the IBM and 4 out of 10 in the Becker muscular dystrophy group did show positive results, definite signs of functional improvement on the distance walked on 6 minutes walking test. So we are going to recruit 3 more patients. We are committed to finish that part of the study. From that point on, we'll look for additional funding to recruit more patients. Would that be answering your question? Okay, thank you.
Okay, we have another session coming in here in a minute. I did promise the last question over there to the right. So after that, I know many of you probably have questions for the medical panelists. They're going to go out into the corridor outside this room because we need to use this room for the session coming up at 11 o'clock. You can find them out there and ask your questions then. So last question over here. She's good. That was nice. Okay. One last round of applause for our medical panel, please. We are back in here for lunch at 12:45.
Okay, I thought